So next we're going to talk about proteins, and we're going to talk about nucleic acids, and these two are going to go hand in hand. You're going to learn later as we talk about protein synthesis and why or how we get these different enzymes and proteins, and how nucleic acids, DNA, RNA, and they play a big role in here. So we're going to talk a little bit about proteins here, and remember that we talked about the polymer. We mentioned polymer and monomers, and the proteins are actually the polymer, okay? And amino acids are, excuse me, I said that backwards. Proteins are, proteins are the polymer, and amino acids are the monomer, okay? So the amino acids are our building blocks, kind of like our Lego bricks. All of these macromolecules that we're talking about, there's a polymer and a monomer, and I kind of picture them as Legos. We're going to build this, great big Lego train with our monomers. So amino acids are the monomer that make up the protein. Okay, and there are 20 different amino acids. And no, you don't have to remember all 20 amino acids right now. But there are 20 different amino acids, and based on the combination that you put these amino acids together, that's what's going to determine what type of protein you have. Okay? And so what makes these different is actually this R group right down here at the bottom. Okay, each amino acid has an amino group and it has a carboxyl group. Okay? And if you look at our 4 little examples over here, alanine, aspartic acid, okay, you will see that they each have an amino group. And I'm going to see if I can do that. There's an amino group. Alright, and they each have, let's just go ahead and they each have an amino group. Okay. And they each have a carboxyl group on the other side. Alright, and what makes them different from each other is actually that R group at the bottom. Okay, and they all have a different R group at the bottom, and this is what makes them unique. Alright, and depending on the combination that you put these together, that's what type of protein you're going to get. Alright. We're going to move over to nucleic acids. DNA and RNA. Deoxyribonucleic acid, DNA, deoxy means 2. Okay? So, what is DNA, or what does it look like? What does it physically look like? It's a double helix. Alright, so you'll see that there's a line here. It comes up around this way. Alright, there's one, and then there's another one that comes all the way down around like this. There's actually 2 strands together, so it's a double helix. Deoxy means, actually, I take that back. It doesn't mean 2. Deoxy actually means deoxygenated. Okay, D meaning taken away. So ribonucleic acid is a ribose sugar, okay, RNA, with an oxygen group removed, and that's a little more detail maybe than what you're going to need to remember. But just know that ribo, means sugar, okay, and deoxyribo is actually a deoxy. It's got an oxygen removed from it, and then it creates this double bond, and it spirals up on itself to make this double helix. Okay, and let's see. So, both of these are made of nucleotides. Okay, you have a 5-carbon sugar, and that's going to be either your deoxyribose or your ribose. Okay, ribose that's a type of sugar. My pen's not working too great here, but ribose is a sugar. Okay, and based on the sugar that you either have ribose or you have deoxyribose. I'm going to have to use my finger. So you either have a ribose sugar or a deoxyribose. Okay? So depending on what sugar you have, that's what's going to determine what nucleic acid you have. Alright, and then there's also a nitrogenous base and a phosphate group. Okay, so these are the three necessary elements to make a nucleotide, and then you string those nucleotides together to get your DNA. So the nucleotide is the monomer in this case. Okay, that's our monomer, and the DNA or RNA are your polymers. Okay, so the nucleotides are our Lego bricks, and we put them all together and make our Lego tower. Okay. So, what is the difference between DNA and RNA? DNA has 4 different types of nucleotides, and let me skip forward just a little bit. Okay? And you don't need to know all of these, the purines or pyrimidines. It's okay to know those. But what I really want you to remember, that there are 4 different types of nucleotides: the adenine, cytosine, guanine, and thymine. Okay, A, C, G, and T. Adenine, cytosine, guanine, and thymine. Okay, and let me skip a few slides here and go forward. Okay, so at the bottom you'll see that how, or right here, how these pair up. Okay, and this always happens. Alright, A always bonds to T, and C always bonds to G. Okay, A with T and C with G. Always. Okay, for our purposes. We get later into genetics, and we get into other types of things. We'll talk a little bit more about what happens if they don't bond together. Okay, so DNA is going to have A, G, C, and T. Okay? But RNA, not only is it a single strand compared to a double-stranded DNA, instead of thymine, okay, so RNA does not have any thymine, it has uracil. So if you remember that A usually bonds with T, but RNA, A is going to go with U, and then G with C. Okay? So there's no thymine in RNA. So it's RNA is single-stranded, and there's no thymine. It's replaced with uracil. Okay, so there's our purines and our pyrimidines, right? Adenine, guanine is our purines, pyrimidines, thymine, and cytosine. What does that mean? It means just the different types of nucleotides. So when you go back here and look at this DNA, they're just slightly different, but those differences can mean quite a big difference later on down the road. All right. So here's and this is more detailed. When it comes to making protein synthesis, and we talk about the DNA is going to unzip. The double helixes with helicase is going to unzip, and then it's going to code for some RNA, which then goes out of the cell or out of the nucleus into the cell, and that's what's going to create your protein. It's the messenger, and we'll talk about that later when we talk about protein synthesis.